Hi, I'm Joe, and I'm a graduate student at the National Institutes of Health. Nowadays, it's common to feel like our lives are being monitored by our smartphones and other technologies. Interestingly, the molecules in our cells are constantly under surveillance as well. One type of molecule our cells need to keep a close eye on is RNA, an essential molecule involved in generating the proteins that our genes code for. My lab specifically studies RNA surveillance, which distinguishes between normal RNAs and RNAs that need to be destroyed because they have mutations that could harm the cell. We don't fully understand how the RNA surveillance system identifies these problematic RNAs, but we do know that it requires a type of molecular motor called an RNA helicase. And one popular model in my field is that the RNA helicase acts like a bulldozer, traveling along long stretches of an RNA and plowing through the proteins attached to that RNA. And this allows it to identify any abnormalities and signal that the RNA is good or bad. However, my experiments suggest this may not be the case. Instead, I found evidence that the RNA helicase may not be so much like a bulldozer, but more like a butterfly, flitting from one RNA to another in the same way that butterflies fly on and off flowers. And this new idea stems from the fact that RNA surveillance could still identify and degrade bad RNAs when I altered the RNA helicase so it could no longer travel along RNAs. On the other hand, RNA surveillance broke down when I altered the RNA helicase so it had trouble jumping from one RNA to another, kind of like clipping a butterfly's wings. My work changes our understanding of the fundamental role for this RNA helicase in the RNA surveillance system. And since RNA helicases are essential for life, it's important to understand how they work, and my research helps us learn more about how RNA helicases keep our cells healthy.